So how does the organizer form? So as it turns out, the cells that give rise to the organizer are induced by two signals. One of these signals gets localized to the dorsal region, and that signal will specify those cells as dorsal cells. And that signal is beta-catenin, which, if you recall, is a transcription factor. The second signal gets localized to the vegetal pole region, and that signal will induce those cells to become mesoderm. And as we'll see later, that signal is a paracrine factor that is known as nodal. Experiments in which different portions of the blastula were recombined show that the organizer is induced by signals that are coming from the underlying endoderm. As we have seen, the organizer is going to form from cells that are located at the marginal zone or the equator, where the animal and the vegetal poles meet. If you were to take an animal cap, which is just the very top of what's going to become the ectoderm, and you recombine that with a portion of the endoderm, this will induce that portion that should become ectoderm to give rise to mesodermal structures, which is what we're seeing in this table. This is different mesodermal structure. Now this shows that the endoderm is responsible for inducing the cells of the mesoderm, including the organizer. Another finding of this experiment is that the type of mesoderm that's induced by vegetal cells, which are the cells of the presumptive endoderm, is going to depend on the region of the embryo from which the vegetal cell is isolated. So vegetal cells that are isolated from the most dorsal side and localized underneath an animal cap will give rise to dorsal mesoderm, whereas vegetal cells that are taken from more lateral and ventral regions will give rise to intermediate and ventral mesoderm and not so much dorsal mesoderm. These dorsal-most vegetal cells of the blastula, which are capable of inducing the organizer, have been called the nucleus center. Evidence for the function of the nucleus center in inducing the organizer came from transplantation experiments using African clawed frogs. Now, in these experiments, scientists took the dorsal-most vegetal blastomeres from one blastula and then transplanted those into the ventral vegetal side of a second blastula. When they did this, the resulting larva had two embryonic axes, which is what you're seeing here. So they had two heads, basically. And similarly, injecting cortical cytoplasm from the dorsal vegetal cells from one embryo into the ventral vegetal cell of a second embryo also led to the formation of a second axis. So these experiments show that these cells that are localized at the vegetal pole, which are the most dorsal, so this is the nucleus center, the most dorsal vegetal cells, this is where the back will form, and these are the cells that induce the development of the dorsal mesoderm. One scientist determined that the dorsal-most vegetal cells, so in other words the nucleus center, could induce animal pole cells to become dorsal mesoderm and as a consequence create a secondary axis. They then set out to determine which factor gave the nucleus center the ability to induce the dorsal mesoderm fate. Beta-catenin was found to be that factor. And in these figures, the beta-catenin is shown in the orange. So this is at the 2-cell stage, and you can see that it's going to be localizing what would become the dorsal-most region. And that localization continues through gastrulation. And recall that beta-catenin is a transcription factor that becomes stabilized by the activation of Wnt receptors. Now, it was found that depleting beta-catenin leads to the loss of dorsal structures, while the addition of beta-catenin into the ventral side of the embryo could induce the formation of a secondary axis. So how does this beta-catenin end up being expressed in the cells of the nucleus center? Okay, so this is where the side of sperm entry ties into the specification of the dorsal ventral axis because the localization of beta-catenin is affected by cortical rotation, which is induced by the entry of the sperm. Now, beta-catenin is initially synthesized throughout the oocyte from maternal mRNA, and then it starts accumulating in the dorsal region of the egg as a result of the cytoskeletal rearrangement that happens during fertilization and early cleavage. Then it continues to accumulate in the nuclei of the cells at the dorsal side, which is what's shown here in part B. And this happens through early cleavage and can be found later at the nucleus center and the organizer regions. Part C shows the ventral nuclei, which you can see do not have beta-catenin. So let's look more closely at how beta-catenin gets into the nuclei of the dorsal vegetal cells and how that relates to the site of sperm entry. Proteins of the Wnt pathway, which include dishevel, which act upstream of beta-catenin, get localized from the vegetal pole of the egg to their dorsal side of the fertilized egg, and that happens during fertilization as a result of the cortical rotation, which is what you're seeing here. These proteins also move along microtubules. Now, two of these Wnt pathway proteins, an example of which is dishevel, act to inhibit another protein known as glycogen synthase kinase 3 or GSK3. GSK3 is a protein that targets beta-catenin for degradation. So by inhibiting GSK3, Wnt and dishevel are going to basically allow the beta-catenin to become stabilized wherever they are found, because they're basically inhibiting the protein that's breaking down beta-catenin. So as a result of that, beta-catenin becomes localized to these dorsal nuclei. And on the ventral side, we have GSK3, but the Wnt pathway proteins are not found there, so GSK3 targets beta-catenin for degradation on this ventral side, and as you can see, there will be no beta-catenin found in these ventral nuclei. So what would happen then if we allowed the beta-catenin to become stabilized not only in the dorsal side, but also in the ventral side? So if we were to do that, for example, by inhibiting GSK3 in the ventral side, what we would get is an embryo that has two axes, which is what you're seeing here. So this is an embryo in which beta-catenin has been stabilized on both the dorsal and ventral nuclei by inhibiting GSK3 throughout the entire embryo. So you can see that we end up with a secondary axis. Now this is the same thing that happens when we take a nucleus center from one embryo and then place that nucleus center into the ventral region of a second embryo. So that provides additional evidence that the factor in the nucleus center that's responsible for specifying the dorsal mesoderm fate is in fact beta-catenin.